Welcome everyone to this Cell and Gene Live. Thanks for being here. We have a terrific presentation lined up for you today featuring our expert panelists, David Dismuke, Ph.D., Chief Technical Officer at Forge Biologics, and Nina Hunter, Ph.D., VP Regulatory and Corporate Strategy at REGENXBIO. Before we get going, I'd like to remind and encourage you to submit your questions for our panelists via the Q&A tool. We will be getting to them during our presentation, so please begin submitting them just as soon as you think of them. We will get to them. Finally, I'd also like to thank Roche Custom Biotech for partnering with us on this event. And so without further ado, let's get started. So AAV vectors have emerged as a leading platform for gene therapy delivery. However, unlocking the full potential of AAV-mediated gene delivery faces some challenges in regulatory, manufacturing, and clinical development aspects, and we want to talk through some of that today. So Nina, I'd like to start with you, please. So based on your experience, what are, would you say, the most critical factors to consider when designing early phase clinical trials for AAV gene therapies?
Yeah, I think there are at least 3 critical factors to consider, and all of these are interrelated. For some of these factors, I think they're even more important when one is considering AAV gene therapy for rare diseases. So one, it's important to think through early limitations of animal models and gaps. For example, these will remain sort of important to think about how they can be addressed so you can best determine how your preclinical studies will translate into the clinic. If there's no appropriate animal model, for example, perhaps in vitro models can be helpful here. So planning that early. Second, related to this translation, it's important to think about best approaches to dose selection. What is the expected biodistribution and toxicology? It's important to understand where the vector travels and think through potential off-target effects. And then lastly, I would say, think about how to best pinpoint your target patient population. What are the appropriate inclusion-exclusion criteria, and what are the endpoints that you want to measure? This is related to disease states, and are these early or late in disease states? Are you trying to reverse the disease progression, stabilize it, and ages of your patients? Because as we all know, there are additional considerations for the pediatric population. And lastly, of course, safety is paramount as it cuts across all these stages. And so ensuring that this mitigation is optimized is very important.
Good, thank you. I want to follow up a little bit on the dose selection portion of what you discussed. How do you approach dose selection and escalation strategies for AAV trials?
This is a question for me?
Yes, sorry, Nina.
Okay.
Yeah.
Thanks. So this is definitely disease specific and indication specific, right? And even product specific. I think there are a lot of learnings from the field, and we now have a lot of clinical experience in the field with specific capsids and routes of administration. We always start more cautiously with dose escalation design here at REGENXBIO. And as an example, if I could give you one, for our RGX-202 program for Duchenne muscular dystrophy, we started with 2 different doses, so 1E14 and 2E14. And preclinically, we learned a lot. So administration here results in a dose-dependent improvements in muscle function in diseased mice, and it led to greater improvements seen at the higher dose. And so at this higher dose, we were able to restore function in the diseased mice in some features. And so these data, along with our clinical experience when we started with 1E14 in humans, led to 2E14 as our pivotal dose that we are moving forward with for our phase 3 study. So I think that's important to think about route of administration and disease-specific situations, where here we believe more vector transduced into muscle cells will likely lead to elevated levels of microdystrophin expression in our case, which are likely to lead to improved function. However, this must always be balanced with safety.
Good, thank you. David, I'd certainly like to give you the opportunity to chime in here as well with your, whether it be with your experience with Forge or perhaps throughout your professional career.
Yeah, so what we see, especially at Forge, we service, we have about a little over 50 clients currently, so we get to see a lot across what people are currently doing. And as far as dose selection, I think what I see a lot is, and I think it's something I urge caution around, is companies really basing their dose their doses on what others have done. And so they see, okay, for this study, whether it's DMD or it's hemophilia or some other, this is what's already being done from a VG per kg basis. And I'd urge, because the titering differences can be pretty profound between groups, that I would, really those preclinical models and how you, like Nina was saying, look at your biodistribution and really being able to, to get widespread distribution and not, I think potentially toxic levels of protein production is something to really consider, and not just try to, gauge it up what others have done in the past.
Sure. Okay. David, while I have you, and I'll certainly be asking Nina for her perspective on this too, from REGENXBIO's perspective, but David, for you, how is Forge leveraging the latest developments in AAV capsid engineering to improve tissue specificity and to reduce off-target effects?
Yeah, what we see is still a pretty profound use of the natural serotypes. Most groups, I think, what we see across the board is probably 75% of the companies we work with are utilizing, the standard AAV serotypes that have been used for a while, because there's a history and it's really well understood how those vectors work, whether it's a systemic administration or a local. But I think probably about 25-30% of our clients are utilizing novel serotypes, and I think this is really, this is an exciting time for the field, quite honestly, where all these Voyager and Capsid and other companies, 4D, have been working on this for a while and really understanding how to do that well, and coming up with better screening and modification strategies, going after specific targets, protein targets on the surface of the cell. So I think it's an exciting time where we can really be able to drop that dose and have a better safety profile by utilizing these novel serotypes. And my suspicion is that within the next 10 years, 5-10 years, I don't know that how many people will continue to use the natural serotypes, because the evolved serotypes can be quite dramatically better than the naturals.
Sure. Okay. And Nina, from REGENXBIO's perspective and your own personal professional perspective, again, latest develops in AAV capsid engineering to not only improve tissue specificity, but also to reduce off-target effects.
Yeah, so at REGENXBIO, we are leveraging specific experience with our NAV technology. Thousands of patients have been dosed with our platform, and we're always trying to build on our deep understanding of AAV vector biology through this platform. And we have developed an optimized capsid engineering platform as well, leveraging multiple approaches from directed evolution to rational design and machine learning to unlock the future of gene therapy with next generation capsids. We have validated our platform and generated improved capsids for various tissues and routes of administration. And as an example, we recently developed suprachoroidal capsids that improve AAV transduction of retina and RPE following suprachoroidal delivery to enable further route of administration implementation. And there are other methods too, such as receptor targeting to access other tissues and try to get cells that are not currently accessible by natural serotypes.